Thank you very much, sir. Hello everyone. My name is Reda Chahir. I am first year PhD student under the supervision of Dr. Naoual Oukkache, which is unfortunately unable due to other commitments today to present. So I present on her behalf the presentation called Development and Characterization of Nanobodies Targeting the Venom of the Moroccan Cobra Naja haje. So for snake envenomation is a major neglected tropical disease. At the global level, there are between 420,000 and 1.8 million snake bite envenomation each year, resulting in approximately 125 deaths per year. This burden is comparable to other major diseases such as meningitis, which causes 28,000 deaths, or measles with 122,000 deaths. Yet, snake bite envenomation is still largely underestimated. Effective treatment critically depends on the efficacy of antivenoms to neutralize severe symptoms. This challenge is therefore at the core of our research focus, driving our efforts to develop next generation antivenoms with improved specificity and potency. Next. After looking at the global and national situation, it is important to consider the regional estimates of snake bite envenomation. Here we see the low estimates, which already indicate a sustainable burden, especially in tropical and subtropical regions. These figures are crucial because they highlight that problem is not evenly distributed across the world, but concentrated in areas where health systems are often under-resourced. Even at the lowest estimates, the impact remains significant, showing that snake bite is a persistent threat to public health in many regions, including Africa. Next. South Africa was for decades the gold standard antivenom in West and Central Africa, targeting 10 viperid and elapid species, including Naja haje. Its production by Sanofi Pasteur was discontinued, leaving a critical therapeutic gap. Although no new antivenoms are now available, clinical data on safety and efficacy are scarce and very few have been evaluated in African settings. Antivenoms are also produced in countries such as Iran, but no data exists on their effectiveness against African snake envenomation. Next. So although some new antivenoms are available, very little is known about their safety and efficacy in African settings. Some products are even manufactured outside the continent, like in Iran, but no data exists on their effectiveness against African snakes. So the first question is, why do we need to evaluate the current antivenoms? And the second question is, why do we need to develop a new generation of antivenoms? Because the ideal product should be safer, more potent, more stable under local storage conditions, less costly and consistently available in the regions most affected. Next. Snakebite envenomation is a growing global health crisis. Current antivenoms are insufficient, leading to high mortality and morbidity worldwide. There is an urgent need for improved, safe, patent, stable, and widely available treatments beyond merely neutralizing lethal effects. Next. Worldwide, snake bite treatments faces major challenges, limited epidemiological and clinical data, a sharp decline in antivenom producers, rising production costs, and complex import procedures. Many existing antivenoms are costly, unstable, can cause side effects, and sometimes have low efficacy. As a result, antivenoms remain expensive and scarce, highlighting the urgent need for safer, more effective treatment. Okay, next. The World Health Organization recommends improving antivenom immunotherapy by adopting innovative production strategies that make manufacturing faster and more affordable. Today, molecular engineering enables the creation of a new generation of antivenoms with superior quality and efficacy compared to traditional polyclonal products, offering better homogeneity, simplicity, lower production costs, higher specificity, and improved safety. So for objectives, next. In this context, our project aims to develop a new generation of antivenoms, specifically targeting the K toxins responsible for severe cobra envenomation. So using advanced biotechnological approaches, we strive to ensure efficient and simplified production, faster and cost-effective processes, high purity and stability, strong specificity and neutralizing capacity. By focusing on nanobody-based therapeutics, our approach leverages the unique advantage of small, stable, single-domain antibodies that can penetrate tissues better, neutralize toxins more effectively, and be produced at a scale using recombinant techniques. Next. On this slide, we summarize the pipeline of our Naja legionnaires venom study. We started with venom collection and initial characterization, followed by fractionation using size exclusion chromatography to separate the components. The most toxic fractions were further purified using HPLC, and these purified molecules were then characterized and tested in mice to study their pathophysiological effects. Finally, we compared our findings with commercial polyclonal antivenoms available in Morocco. This pipeline provides a clear understanding of the K toxic molecules, which guides the design of our next generation nanobody-based antivenom. Next. Let me walk you through the three main work package of our NGA project. First, we focus on identifying and purifying the K proteins responsible for the most severe symptoms in inflammation. These proteins serve as our candidate antigens for the next generation antivenoms. Second, we perform dromedary humanization to stimulate their T lymphocytes, which are essential for production the nanobodies or VHH. And finally, we move to VHH production, creating small, stable, and highly specific nanobodies. On the following slide, we will present the methodology we used for our work package one. So next, for the purification workflow of Naja legionnaires venom involves venom collection, initial characterization, toxicity, pathophysiology, and mass spectrometry, fractionation by gel filtration chromatography, characterization of the purified fractions, further fractionation of toxic fractions by HPLC, and final characterization of HPLC peaks using toxicity assays, electrophoresis, and mass spectrometry, identifying then the target fractions. For the results, next. Identification. So on the following slides, we will present now the results we obtained for this work. So next. So for the lethal dose 15, values indicates that the venom is extremely toxic, primarily due to low molecular weight toxins that rapidly enter the bloodstream. Gel filtration chromatography of Naja venom yielded 8 fractions of with fractions 5 and 6 accounting for 85% of the total venom. Next. Electrophoretic profiling of the whole venom and its fractions revealed that fractions 5 and 6 are mainly composed of low molecular weight proteins, approximately about 40 kilodalton. Next. Fraction 5 contains proteins of various molecular weights, but these proteins are either present in very low percentage or completely absent. This indicates that fraction 5 is enriched in specific components, while other types of protein are minimally represented or missing. Next. Fractions 5 and 5/6 retained most of the venom's lethal activity. For the fractions 5 only induced 100% mortality within 5 minutes, causing neurotoxic signs and loss of reflexes. Other for fraction 6, caused toxicity without death. Other fractions 1 to 8 to 4 and fractions 7 to 8 showed weak or no lethality, producing only mild signs like hypoactivity or transient ataxia. Next. Using this workflow, we can accurately identify the proteins present in the toxic fraction, including fractions 5 and 6. The trypsins digested peptides yield N-terminal sequences, which are then compared to protein databases using BLAST. And this allows us to assign each protein to a known protein or protein family, providing a detailed map of the molecular components responsible for toxicity. Next. Our results show that the toxic fraction of the venom is composed of 93% of toxins and 7% other peptides. Breaking down the toxic profile, we found cytotoxins at 53%, neurotoxins at 45% percent, and cardiotoxins at 2%. It's important to note that cardiotoxins, although present in small amounts, are considered highly potent and relevant for toxicity. Next. In the toxic fraction, we identified different types of cytotoxins, with cytotoxins 5 being the most abundant. For the neurotoxins, the profile includes long neurotoxins at 13.6%, short neurotoxins at 9.4%, and other neurotoxins. Next. For fractions 5 was further fractioned by HPLC, and the resultant peaks were analyzed by mass spectrometry to identify the specific protein component present. Next. Our results show that fraction 5 is composed of 63% cytotoxins and 36% neurotoxins. Breaking down the toxic profile, we have cytotoxins at 53%, neurotoxins at 45 toxins. 45%, sorry. Next. Also, for fraction 6 was fractioned by HPLC, and the resultant peaks were analyzed by mass spectrometry to identify the protein components present. Next. Our results show that fraction 6 is composed of 77% of cytotoxins and 11% of neurotoxins, and 19% of neurotoxins. Within the cytotoxin profile, cytotoxins 5 is the most abundant. Next. For the most toxic gel filtration fraction, is the fraction 5 was further purified by RPHPLC, producing several subfractions. Among these, SF3, the subfraction 3 derived from like she's derived from the peak 3 was highly lethal with a lethal dose of 1.62, causing severe neurotoxic symptoms and death. Other subfractions, such as subfractions 16 and 19, were no lethal but induced moderate local cytotoxic effects. So these results indicate that the lethality of Naja haje venom is mainly due to specific hydrophobic subfractions of fractions 5. While other components contribute to tissue toxicity. Importantly, these lethal subfractions like subfraction 3, 16, and 19 serve as candidate antigens for the production of next generation antivenoms. Next. Mass spectrometry results revealed that peak 3 correspond to short neurotoxins. Next. Results of mass spectrometry show that peak 16 corresponds to cytotoxins 1, 2, 3, 4, 5, and 10. And on the other hand, next. And on another hand, the results of mass spectrometry shows that the peak 19 are also composed by cytotoxins 1, 2, 3, 4, 5, 9, 10, and 11. So next. Having identified the key toxic fractions and the candidate antigens in Naja haje venom, we can now move to next stages of our project. The focus shifts to work package 2 and 3, where we'll leverage these antigens to develop nanobody-based antivenoms. Next. About 6 weeks after camelid immunization, with repeated injection of fraction 5 and fraction 6 venom fractions, blood is collected, B lymphocytes are isolated, analyzed to extract total RNA, including EGJ, mRNA, complementary DNA, cDNA is synthesized by reverse transcription, and nanobody sequences are cloned into a fragment to generate a library representing the full heavy chain antibody repertoire. The library is enriched by phage display biopanning against the antigen. Unique specific clones are selected by ELISA and identified by sequencing. Selected clones are subcloned into an expression vector expressed in XPy cells and purified by affinity chromatography, typically followed by thick chromatography. Next.
Reda, 5 minutes left.
Okay. B lymphocytes are collected from the immunized camelid, and RNA is extracted and reverse transcribed into cDNA. A two-step PCR amplifies the VHH sequences, which are ligated into the 4.5 VenOx fragment factor for expression as P3 fused nanobodies. Next. And in the following slides, we will present now the results we obtained for this work package. Next. RNA quality and integrity were assessed before library construction, and electrophoregrams showed clear RNA peaks, as you see. So these confirm that all samples are of sufficient quality for tRNA library construction, with samples 3 and 4 showing slightly higher integrity. Next. tRNA was reverse transcribed into cDNA, serving as a template for PCR amplification. In the first PCR1, 5 primers amplified 2 fragments, confirmed by gel electrophoresis, and second used 3 primers to amplify VHH fragments and introduced like the 2 SFL and Naoual restriction sites for cloning. Next. After generating the VHH gene fragment by PCR, they were cloned into the VEN6 fragment vector. This vector fuses the VHH to P3 phage protein for display and contains PEP for perisplasmic secretion, as you see. Next. To assess the quality and diversity of our library, 24 colonies were randomly selected and grown. Overnight, colony PCR with vector-specific primers confirmed correct fragments insertion as shown by clear uniform bands on the shell. Sequencing of the clones highlighted variable regions, including the CDR, and analysis indicated 100% library diversity. But next. And during library biopanning, we used the toxic subfraction of fraction 5 as antigens to enrich high affinity nanobodies. The subfraction 3, 6, 16, and 19 were used as antigens. Next. Before we begin biopanning, let me show the workflow for preparing and expression the VHH phages. After each selection round on antigen-coated ELISA plates, isolated colonies are expanded in primary cultures and then in secondary cultures. Next. 3 rounds of biopanning were performed to enrich phages with high affinity for the target of peak 3. Next.
1.5 minutes left, Reda.
Okay. So 3 rounds of biopanning were performed to enrich. Next, please. 3 rounds of panning were performed to enrich phages with high affinity for the target P16, peak 16. And next, at least 15 different nanobodies were identified against peak 16 of Naja haje legionnaires venom, with sequences found more than 20 times highlighted in color. And next, after biopanning, at least 6 major VHH sequence families were identified for subfraction 3 and 16 with Andra cluster identity below 90%. These clusters show in the slide group nanobodies clones based on sequence homology and antigen specificity. And next, 6 sequences compressing neurotoxins and cytotoxins were analyzed and showed notable homology. Next. Sanger sequencing confirmed that 6 of 8 clones matched expected VHH profiles, alignment and clustering, especially of CDR3, revealed redundant sequences and indicate selective enrichment for specific epitopes. Next, please. Of the 6 clones studied, only clones 4, 5, 4 and 5, 7, and 14 showed successful affinity chromatography results with distinct peaks indicating effective binding to the protein G colon and successful VHH purification. Next, in contrast, clones 2 and 6 showed no significant peaks during affinity chromatography, suggesting low VHH expression or potential binding issues with the colon. Next, please. So for the SDH page analysis confirmed a single approximately 55 kilodalton bond for successfully purified nanobodies, indicating their expected size and purity. Next. For fraction 5, clones 4, 5, clones 4, 5 and 14 exhibited strong specific binding to the antigen as indicated by classical sigmoid curves, whereas clone 7 showed a slower increase, suggesting lower affinity or delayed saturation. Next, please. Neutralization assays of toxic activity with the 2 types of antivenom showed that the antivenom based on nanobodies we developed neutralized mortality with a potency 4 times higher than that of commercial antivenom Inoserp. Next.
Can you wrap up, Reda, because you have minus 1 minute left.
Okay. Next. No, before, please. Yes. Histological evaluation at 48 hour post-injection revealed normal brain architecture and controls, as you see, while fraction 5 induced severe neurotoxicity with neuronal degeneration, edema, and inflammatory infiltrates. Nanobody treatment markedly preserved neuronal structures, reducing edema and inflammation, whereas commercial antivenom offered only partial protection. Next. As for VenOmics provides powerful insights into venom complexity, identifying K-toxins and guiding the development of targeted therapies. Considering inter- and intra-specific variability is crucial to ensure effective and locally adapted treatments. The development of adjuvant can enhance antivenom efficacy, while recombinant nanobody-based antivenoms represent a promising next-generation therapy due to their high affinity, stability, and specificity. And next, and thank you for your attention.